Hi, this is Dr. Tuba Semolu from Üsküdar University Department of Bioengineering. Today I would like to present you with a topic that we prepared with Özlem Arena'nın. It's the investigation of the association of human antimicrobial peptides and human intestinal microbiota through bioinformatics tools.
Antimicrobial peptides are essential components of innate immunity in humans and other higher organisms, and they contribute to the first line of defense against infections. They are produced by intestinal epithelial cells, and they play an important role in the regulation of intestinal homeostasis by regulating the microbiota.
Let me explain briefly what metabolites are. Metabolites are small molecules and they're intermediates and products of metabolism. They play important roles in living organisms, and there are two kinds of metabolites. The ones that are required for growth and maintenance of cellular function are called primary metabolites, and the ones that are not required for growth and maintenance of cellular functions, and they are the end products of the primary metabolism, are called secondary metabolites. Especially secondary metabolites are compounds of diverse types of structures, and they serve as weapons against bacteria, fungi, plants, and insects. Secondary metabolites are also used in signaling pathways. Metabolites are useful for fuels, medicines, cosmetics, food supplements, ecology, and healthcare. The immune system is a defense mechanism in the body that includes T cells and various blood cells derived from bone marrow, such as B cells, macrophages, and dendritic cells. The function of the immune system is to eliminate infectious microorganisms that invade the body and cancer cells produced by mutations. Immune reaction leads to cell death under certain conditions. Therefore, excessive elimination of targets in chronic inflammatory reactions is harmful and is the cause of autoimmune diseases. Let me talk about human intestinal microbiota. The human intestinal microbiome is among the most complex of the body sites. It includes 500 to 1,000 species and several million genes. There is a complicated bidirectional relationship between the intestinal microbiota and the host, which is likely critical for human health as well as implicated in disease pathogenesis. The gut microbiota is important in digestion of food and extraction of nutrients, modifying the host immune response, protecting against infection, the metabolism of drugs, participation in and regulation of host metabolism. In this study, the connection between human AMP microorganism and human microbial metabolite microorganism was examined with a systems biology approach. Here, I would like to talk about the method of the study. Firstly, AMP databases were determined. Then 123 human AMP data were selected from the antimicrobial peptide database, and also 148 human microbial metabolite data sets were selected from the human metabolism database. And from these lists, microorganism AMP and microorganism metabolite relationships were determined. The AMP gene symbols were obtained using BioDBNet, and then the enrichment analysis was done using EnrichR bioinformatics tools. The visualizations were done using Cytoscape, and we used BioSeq database collection to obtain the KIG IDs of metabolites, and metabolic pathways were obtained using EMBRL2 database. Here you can see the association of the antimicrobial peptides and microbe species that were obtained using two of the databases that I have mentioned before. In this slide, you can see the visualization of antimicrobial peptide microorganism association. And in figure B here, you can see the hubs of the association. Basically, there are three antimicrobial peptides that were most interacting with the microbe species, and the most interacting microbe species can be seen here as well. In this slide, you can see the association of metabolites and microbe species, and also the KIK IDs of the metabolites. Again, we did this using the databases that were given before. Here you can see the association of microorganisms and metabolites, and also the hubs of the association. We can see the enzyme, the metabolites succinate, acetate, and formate being the ones that are most interacting. We can also see some of the microbe species that have been interacting with the metabolites. In the previous slide, I had mentioned about succinate being the hub of the association of metabolites and microorganisms. Succinate is a Krebs cycle metabolite, and it accumulates in macrophages during inflammation, and it's shown as a signal to activate pro-inflammatory gene expression. Also, in multiple sclerosis, succinate functions as a pro-inflammatory mediator in the central nervous system upon its release from cells. Enrichment analysis was done to obtain information about the AMPs. For instance, here you can see the cell type locations of AMPs. Some of them are in bone marrow, some of them are in salivary glands, and some of them are in small intestines, and some of them are in tongue. Here you can see the signaling pathways associated with the AMPs. Chemokine signaling pathway and cytokine receptor interaction have been shown to be statistically significant here. As the main regulators of cell migration, chemokines and chemokine receptors are critical for basic biological processes such as embryonic development in early life, autoimmune diseases, host defense, and wound repair throughout life, and age-associated diseases. The antimicrobial peptides were also enriched in gene ontology terms. One of the gene ontology terms is biological processes. Here you can see the processes such as defense response to bacterium, lymphocyte chemotaxis, granulocyte chemotaxis, and response to interleukin-1 as being statistically significant in this enrichment analysis. The antimicrobial peptides were enriched in another gene ontology term, cellular component. Here we can see that the statistically significant terms were secretory granulomin, specific granule, and mostly of granule, tertiary granule. The other gene ontology term that the enrichment was done was biological processes. Here we can see chemokine activity, cytokine activity, copper iron bindings, cysteine type interpeptidase inhibitor activity, and G protein coupled receptor binding. We also examined AMP disease association. Here we can see that dementia, osteoporosis, and lateral sclerosis are all associated with antimicrobial peptides. ALS is a motor neuron disease. Dementia is any decrease in cognition that is independent, important enough to interfere with daily function. Osteoporosis is the most common metabolic bone disorder that affects 200 million people worldwide. In conclusion, in this study, our analysis indicated that there were 2 AMPs mutually associated with 6 microbe species that I have mentioned here, and also 4 of the intestinal bacteria, including E. coli, share a mutual metabolic pathway, which is the succinate metabolite through metabolites. The AMP list was also enriched in signaling pathways such as chemokine signaling. And here, by establishing a link between human microbial metabolites and human antimicrobial peptides, the possible connections between AMPs and diseases such as diabetes, psoriasis, rheumatoid arthritis, multiple sclerosis, dementia, and osteoporosis may be eliminated. Future studies investigating the molecular mechanism of diseases should include microorganisms and metabolites in humans for a better understanding of such disease. I want to thank you all for listening and have a great day.